About the history of microscopy, you can see that there are different types of microscopes here, which was like an early English microscope in 1680, and the other one was made in 1800. The idea that all living things are composed of cells developed over many years and is strongly linked to the invention and refinement of the microscope. Early microscopes in 1600s opened up a new field of biology, the study of cell biology and microorganisms. Some of the first organisms to be studied were the single-cell planktonic organisms of pond water. And also we are going to study these pond water as well, and we will going to take the sample from the lake and then we will observe the microorganisms under the microscope. And this beautiful paramecium is one of them. We were supposed to find this paramecium when we take a sample from the water. Early microscope had some specific structures like the single lens, specimen holder, and a place for focusing and adjustment, and the screw thread. Anthony Van Leeuwenhoek designed and built over 500 single-lens microscopes. They provided a magnification of 270 times. Bacteria, protozoa, human blood cells, and spermatozoa were discovered using them. By today's standards, the Leeuwenhoek microscope in right side both of them are regarded as glorified magnifying glasses. And here is the Robert Hooke telescope. Robert Hooke described the use of a compound microscope he devised in 1665. Sorry, Robert Hooke microscope, I said by mistake, telescope. So Robert Hooke described the use of a compound microscope at the right side in 1665. He was the first to coin the name cell after he observed the angular spaces that he saw in a thin section of cork. And here is the compound microscope. We have exactly the same thing in our lab. The compound microscope is a fundamental tool in biology. High-power light microscopes are used to magnify objects up to 1,500 times. They are called compound microscope because there are two or more separate lenses involved. Why do we say separate lenses? You can see over here. There are two types of lenses. Thin and mostly transparent samples are required so that the light from the microscope can pass through the sample easily to reveal the details. So here is the eyepiece lens. Then there is an objective lens. A mechanical stage, so you can move this mechanical stage left and right by using these, and also these. This is a condenser, inbuilt light source. You can increase or decrease the level of the light by changing over here. The focus knob, this is what I said, so you can use it for going movement of the mechanical stage it can be done using these. And the coarse focus knob that they can be used for going up and down. So this stage goes down and can goes up while you are touching this. And here is the arm. So here is another type of microscope for dissection or dissecting. The difference between this microscope and the previous one is the magnification also. And also you can put the very large and big samples over here because this is used for dissecting. Dissecting microscopes such as that shown right are the type of binocular microscope. So you can see using your both eyes, which is called binocular. They are used as a low magnification, which means that 4 to 50 times. So they can magnify the objects or the samples until 4 times, from 4 times to 50 times, not more than that. A dissecting microscope has 2 separate lens systems, one for each eye. And a 3D view of the specimen is produced. So if you use your both eye, then you can see the specimen very clearly using both eyes. Because of this, they are sometimes called stereo microscopes. So here are the different types, different parts of the dissecting microscopes: eyepiece lens, height adjustment knob. So you can go up and down. So by using these, you can go down or up. So up and down. And this one, which can be used again for movement of the the objective lens, and the stage that you can put your samples over here. And this is the eyepiece focus. Okay, what does magnification mean? Here is fly. So in this picture, this is the real or actual size of the fly. What happens when you magnify? So here there is a magnification by 5 times. So of course you will see 5 times more comparing to the actual size when it is written x5. And what happens if you observe 25 magnified? So you will see that the magnification is completely higher than this one. And here, if it is the 50 times more magnified. So magnification refers to how much larger a sample appears to be compared to its actual size. As magnification level increases, so does the blurriness of the sample. What is resolution? Resolving power or resolution is an important factor determining the usefulness of a microscope and is a measure of the clarity of the image. It is minimum distance, 2 points can be separated and still be distinguished as 2 separate points. For example, what appears to the unaided eye as 1 star in the sky may be resolved as 2 stars with the use of telescope. Electron microscopes have a greater resolution than optical microscopes because electron beams have wavelength much shorter than those of visible light. Examples of high, medium, and low resolution for separating two objects viewed under the same magnification are shown by. So you can see them here. This is the higher resolution, with complete and very clear. This is a medium resolution, and here is a low resolution. The next topic is bright field microscopy. Here are some, the background. It is explaining the backgrounds of the microscopy. For example, here is bright field. The bright field lightning reveals very little details unless a cell is stained or naturally pigmented. Yeah, here is a bright field. So if it is a bright field, the information that you can get is less and not that much. And cell walls and nuclei may be visible, but not the other type of information that you need from the cells. But once you talk about dark field, in dark field, illumination is excellent for viewing near transparent specimens. So if the specimens are transparent, so you can see in the dark field very much better. And the nucleus of each cell is visible. So with a conventional bright field microscope, we see objects in the light path because natural pigmentation or strains absorb light differentially, or because they are thick enough to absorb a significant amount of light despite being colorless.